Thank you, Edward. So what I'm going to do is I will have the fun presentation. The last one's always the fun one. It's the quick wrap up where we get to actually look at some fancy technology and especially leave the kind of symposium with some food for thought. So hopefully I can do that for you today. So as Edward said, essentially what we're going to discuss is patenting tissues for actually cell microenvironment engineering. But while I will take one step to acknowledge Professor Hoffman's very excellent introduction to this type of technology. The whole field is generally known as hydrodynamically confined flows, where we can control the liquid environment external to the microfluidic device. And what that really empowers you to do is deliver compounds, as we've seen a few times. But also, one thing I will show in a few slides is the ability to deliver things which are within the flow. So we can actually use it to bioprint cells and actually build the microenvironment that we want to probe with our single cell experiments. So I actually just grabbed this slide from an old presentation just to give Professor Hoffman an updated set of images on things. So we can actually show, how the hydrodynamically confined environment, changes, in real time. So we can see these very strongly fluorescent compounds actually changing. What we have here, or as in these two zones, especially on the right side, is demonstrating one liquid being introduced into another one, and there's no mixing and there's no loss. However, when we are dealing with these hydrodynamically confined flows, we want some sort of validation that, okay, if we're going to introduce liquid A into liquid B, we don't get lost to the surrounding area. This is very important when you're using agonist antagonists or doing any sort of electrophysiological readings. You want to have confidence in your platform. So one of the things we equally looked at recently for another reason was to actually set up a continuous recirculation zone, which is shown in the bottom, and then look at different distances away from the tip. And then by probing these different distances, we're able to actually see if any of that compound actually enters that. So we use very long exposure time on the cameras to actually see, does any leakage occur? And what we're happy to say is that yes, we actually do maintain a confined zone at 10-second continuous exposures, even 30-second continuous exposures. What we see is we go back down to background every time. We don't have any transient solutions being lost into the environment, and we can do this repeatedly. What you also get from this is, by studying the hydrodynamically confined zone, we can see that we're actually getting fairly square wave activation. We get very sharp on and off times. And when you're looking at compound delivery for studying these kind of things that have been described today, looking at ion channel responses, it's very good to be able to decouple the liquid delivery from the actual response of the ion channels or the biology itself. So having these on-off times as square wave-like as possible allows you to have some sort of a decoupling of the biology from the liquid delivery as a whole. One particular example, because I have to show something different that has been seen before, is actually a project that was done a few years ago, actually studying communication within muscle fibers, actually looking at what sort of interconnectivity there are within mitochondrial networks within one particular fiber. And here we can see that we're delivering to one part of the fiber, and it's not spreading throughout, and we're not losing anything to activate larger parts of the fiber. But what I've shown so far is exactly what has been kind of raised by other people, which is this approach is very good at delivering liquid to a small area. It does so in a contamination-free manner. It does so with very little volume, and it does so with a very high repeatability, both on the area being exposed and also the timing of what's being exposed. However, this can be expanded. So as I was elucidating at the start, if we can control the liquid handling within one small region, can we use this laminar nature to actually deliver things other than solutions? So can we put something in there, such as cells, to actually micro-pattern the environment that we want to study if we're interested in how particular cell networks work, or if we're interested in what the change if the local environment is on the response of a particular signal or a response for a particular cell. Can we build that microenvironment using this approach? So we developed essentially a single cell bioprinter where we can use the power of microfluidics to position not only the printhead, but the things which it contains, in this case, it being cells. This can work in 2D and 3D, but one of the big separating factors of this technique, particularly relevant for electrophysiology or other probing techniques, is that this can be done in the liquid environment. We don't need to have anything covering the cells. In other words, as soon as the cells sit down and are happy and maybe stretching out, they can then be interrogated directly. You can use external probes. They can be connected with patch pipettes, for example. So this is very amenable to other electrophysiological and probing techniques. And like Professor Hoffman was indicating earlier, the volume usage at these sort of size scales is incredibly small. So it's very good for working with very scarce or very valuable cell samples. So just to give a little patterning example, just to show kind of what this kind of control can empower you to do, is here's just a typical pathological skin slice, just to show how cells can be patterned within a sample. And we're going to take that image, and then we're going to pattern cells in exactly the same way. So here's a little video of us essentially building that type of patterning, whereby we try to replicate the gap regions, the curvature which would be there in the histology. We try to replicate the local environment, the density of cells which are in the different areas, so that we can try to establish a system which better resembles what the cell might feel in a native environment. So what we can do is take a blueprint, it being either from a histology sample, or we can take a blueprint from something which is in research, or in fact something which is just of interest, i.e. a particular ratio of cell type A to cell type B. This can be then built, printed, and then it can be studied in however we want to. The one advantage, as I was mentioning, is that because we use no binding matrix, we can put cells on top of other cells very, very locally. And we can do this in both 2 dimensions and 3 dimensions. The kind of reason or rationale for that is if you need to really control the microenvironment. So if we need to really eloquently control the microenvironment, here being an example of a study for studying stem cell and cancer cell interactions. These are actually done on top of other cells, like seeding layers of cells, where we can pattern very precisely the different regions, so we know what the kind of contact area between different types of cells are, and we know effectively the ratio difference between the cells, so they each are in their own microenvironment. This can be done with many different things, as you can imagine. Whatever we can load into the fluidics, we can print and we can pattern. Sometimes it's more interesting to work in 2D models. Sometimes it's 3D models, as we can see in the top, both with skin. On the right side, there's a malignant melanoma model. But sometimes it's more interesting to actually just control the local environment. So we've done projects and work with people on actually patterning the local microenvironments of stem cells, these being MSC stem cells. And on the bottom right, we can equally see printing and patterning and actually allowing primary neurons to form processes, to form connections to other cells. So these can all be essentially studied so that you then have a patterned system that you can then interrogate using other electrophysiological or fluorescence techniques, this allows you and then gives you the control to build your system, as opposed to just allowing the cells to be where they are. So just as a kind of quick wrap up, what essentially HCF or hydrodynamically controlled tools allow is this ability to deliver compounds and control the microenvironment around these individual cells. This control of microenvironment can in fact be building cells around the cell that you're actually of interest in, which allows you to then build some form of microtissue from the bottom up, from individual cells. Allowing you to be able to do this means that you can get repeatable environments. So you can actually replicate the environment that you are most interested in from a single cell perspective, giving you this ability to have microorganization of the tissues, to study cell-to-cell interactions and how that actually changes or has impact on the measurement system, how maybe critical cell densities or maybe the inclusion of rare cells into the sample can affect the responses of various cellular systems. And also because we still are founded on this solution exchange, we can then go to these tissue systems and directly control the microenvironment using the solution exposure system. So, and with that, I will wrap up and I will stop sharing and I will be open for any questions.
Thank you very much, Gavin. This presentation is amazing. I mean, for everyone that work with single cells and want to create tissue model and disease model and any type of pattern. Is there any questions from our speakers? Feel free to ask. Yes.
Yeah, I would be interested in how shear forces would affect the viability of cells when being printed. I mean, it surely depends on the size of the cells, because the size of the channel the cells go through is constant.
Yeah.
So
yeah.
What do you know about this?
Yeah, so it definitely, I mean, it's always a concern. I mean, I would with regards to, and I can show if, or I can make available the cell types, you know, that we have worked with. We don't notice any negative effect on the viability or the survivability. So we recently published in scientific, Nature Scientific Reports, using this device to actually do bioprinting constructs in 2D and 3D, and actually looking at basic like liver model systems, and whereby we actually, for all the systems we did, we measured actually the viability compared to standard cell culture and handling. And what we actually saw is that it's either identical or, you know, approximately better. I mean, because it kind of positively selects for the healthy cells, because the healthiest ones stick better than the not healthy ones. So you actually can, the cells which would be within the circulation that maybe not as healthy as they should, don't stick as efficiently. So then you get the most kind of bang for your buck, if you want to say, when you're actually printing and patterning the cells. But from our kind of data, we're in the above 90%, and in some cases 95%, both viability and survivability, which has been checked.
Thank you.
Yes, Professor Hoffman, you can talk.
How long does it approximately take someone to train on this machine to really use it efficiently?
It's a very good question. I mean, one thing which we wanted to do from the start was taking actually experience from feedback from people like yourself that actually have used the BioPen, is to remove as much fluidic understanding as necessary as possible and actually switch it to just biology. So we actually use everything on a gamepad. We use a little joystick controller, and you just move around. I mean, you can use the kind of CAD type interface and you can draw what you want, or you can just run it in free run mode and actually just translate where you want. I would say that the only, let's say, hurdle to printing is being able to position, you know, a device. it does have a series of different automated, like, focusing and everything else, like a lot of platforms do, but you still need to have some understanding of positioning a probe. But with regards to training somebody, I mean, within half a day, I mean, you can be printing fairly comfortably. But as I'm sure you're aware, handling of the cells and the biology, that's where the difficulty comes, not the actual operation of the machine.